Good afternoon, dear viewers. Welcome to CEC EduSet live lecture. I am Dr. Amit Bhattacharya from Department of Zoology, Ramjas College, University of Delhi. And today I will be speaking about the antimalarial drug resistance, the basics and the emergence. Before going into the drug resistance part, I will just give you a brief overview what exactly these vector-borne diseases are, how they are transmitted across person to person, what are the various diagnostic measures which are available, and then the antimalarial drugs which are available. Now, most of these vector-borne diseases are transmitted by the vectors. Vectors are basically the living organisms that can transmit the infectious pathogens between the humans or from the animals to the humans. And these vectors are generally blood-sucking insects. So we can see some of the vectors over here, the mosquito, bed bugs, lice, tick over here. So these, what they do is they ingest the disease-producing microorganisms during their blood meal and then transmit it to a person who get infected. So that's how the cycle of infection goes into the picture. Now, the vector-borne diseases has been recorded as one of the major infectious diseases by World Health Organization. Now, these causes the human illness basically because of the transmission and the development of the parasites, virus and the bacteria within the host system, and they are transmitted by the vectors. Every year about 7 lakh deaths from these diseases occurs, which mainly are from malaria, dengue, African trypanosomiasis, leishmaniasis, Chagas disease, yellow fever, Japanese encephalitis. Now, we come into the antimalarial drug development programs. Now, the malaria we know is transmitted through the bites of the infected female Anopheles mosquito. Of the 400 different species of Anopheles mosquito, only around 40 are malaria vectors of major importance. There are different species of Anopheles mosquito which have got their preferred aquatic habitats. Now, these Anopheles mosquito complete their life cycle in 3 forms. So, what happens is the eggs are laid, then they hatch to produce the larval form, which undergoes several molds stages before they emerge out as the pupal stage, and finally the adult mosquitoes are formed over here. So, we can see the Anopheles mosquito cycle majorly occurs in the water presence area. So, the stagnated waters plays a very important role in the development of these mosquitoes. Now, some of the key facts which have been noted down by the WHO is the vector-borne diseases accounts for more than 17% of these infectious diseases, causing 7 lakh plus deaths annually. And they majorly are caused by the parasites, bacteria or the viruses. Now, in this one, the malaria is one of the major causing agents which in the globally seen, if we see, there are 249 million cases which are being reported, of which about 6 lakh 8,000 deaths occurs every year, and it majorly affects the children under the age of 5 and the pregnant woman. Now, another the vector-borne disease is the dengue. So, dengue is a prevalent viral infection which is being transmitted by a different class of mosquito which comes under Aedes type. More than 3.9 million people in over 132 countries are at a risk of contracting dengue, and about 96 million symptomatic cases, an estimate of 4 lakh deaths occurs every year because of the dengue infection. Apart from that, there are various other infectious diseases which are there, the vector-borne diseases, which includes the chikungunya fever, Zika virus, yellow fever, West Nile fever, Japanese encephalitis, to name a few. Now, this is an image which is showing the major vector-borne diseases over there. So, we can see the malaria, leishmaniasis, Japanese encephalitis, chikungunya, dengue and filariasis are the major vector-borne diseases which compromise the various vector-borne diseases transmitted majorly by the vectors over there. Now, the main vectors and the disease, how they are transmitted, are majorly by the various mosquitoes or the related insects organisms. So we know that the Aedes transmit the dengue fever, yellow fever, chikungunya. Anopheles transmit the malaria, while the Culex transmit the Japanese encephalitis, lymphatic filariasis, West Nile fever, to name a few. Then comes the various ticks and the sandflies. So the sandflies, one of the major disease is the sandfly fever. Then the ticks transmit the Lyme disease over there and the various tick-borne encephalitis diseases. Then there is a tsetse fly which causes the sleeping sickness, fever over there. Fleas and bugs also causes number of disease conditions. Now, this is a diagrammatic representation which is showing how the vectors transmit the parasites into the host system, and there are various different parameters which plays an important role. For example, the movement or the migration of the individuals, poverty is one of the thing, deforestation is one of the major factors. So all these factors are interlinked to one another and they lead to the transmission of these vector-borne diseases across the population. Now in India, if you want to know more about the vector-borne diseases, you have to log into the National Center for Vector-Borne Diseases Control Program website. So this is a screenshot of the main page of the website which shows the various disease conditions, and if you click on the tabs which are present, you get to know about various vector-borne diseases. Apart from the knowledge of the vector-borne diseases, it gives you a reference of the statistics as well as the various treatment and the diagnostic measures which are available for these particular diseases. Now, in the malarial case, if we come back, the key facts which are there by the WHO is, if you see in the 2023, there were estimate of 263 million malarial cases which were reported from 83 countries. And of these, the maximum number of cases are being reported by the WHO African region, which proportionates a high stake of these global malaria burden cases. And this African region constitutes about 94% of the malaria cases, which comes to about 246 million cases and 5,69,000 malarial deaths. The children under the age of 5 are mostly affected in this particular region over there. Now, every year the World Health Organization releases a brochure with all the guidelines, statistics and the various treatment and the diagnostic results, and this is called as the World Malaria Report. So I will show you some of the important statistics and the reports outcomes in the World Malaria Report which have been published in the year 2024. Now this is a diagram which is showing the proportion of the malaria cases across the different parts, and if we see, the largest red dots are from the African regions. As we just discussed in the previous slide, the African regions are the one which constitutes the major number, or about 95% of the cases come from these particular regions. Now this is a diagram, the map which is showing the spread of the malaria cases. So the red shows the maximum number of malaria cases coming from those particular regions. So we can see the African region, South American region, the Asian region including the India are the major reporting the major number of malaria cases across the various states and the districts. Now the WHO Global Malaria Program was released, which was having 4 key roles into them, which included how the malaria diagnostics has to be done, then the treatment has to be done, and finally to counter check how the treatment has gone underway, because it is very essential to know how the treatment is being done so as to avoid the malarial drug resistance over there. Now the global technical strategy which has been released by the WHO, which is called as the GTS strategy for malaria 2016 to 2030, has got various pillars. So there are 3 main pillars which are there. Apart from that, there are adjacent pillars which are there. So each of these pillars, if you see, speaks about how exactly the diagnosis is being done, how it is located, what are the programs which has to be characterized for those particular areas, and based on those programs how the treatment has to start, and subsequently the surveillance of these particular treatments has to be made. So these are playing a very important role in decreasing the number of malarial cases across the globe. Now, the WHO guidelines for the malaria, it states that nearly half of the world population is at a risk of malaria, and in these high malaria transmission areas, especially the young children and the pregnant women are the particular ones who are most vulnerable to these malarial infection and the death cases. So it is very essential to stop these cases at the level of its beginning. Now, since 2000, there is a expanded access to the various World Health Organization malaria prevention tools and strategies, which are being divided into two major forms. One is the effective vector control programs, the other one is called as the preventive chemotherapies program over there. So the vector control program, as the name is suggesting, it means that the WHO is giving the guidelines to control the vectors, which is mosquitoes. Now, in the preventive chemotherapy, it means that the use of the drugs and the various antimalarial drugs which are being released into the market or are present for the clinical uses are being used to reduce the global burden of this disease. Now, this vector control measures is broadly divided into two major parts. One is called as the long lasting insecticidal bed nets, and the other one is the indoor residual spraying. So we will see each of them one by one, and finally the next one comes is the chemotherapies, which means the use of the antimalarial drugs to cure the patient. Now, the vector control program guided by the WHO has been intervened into two major forms. One is called as the ITN, which is the insecticide tested bed nets, which is being done. So there are various pre-qualified bed nets which are available and are in the market, which are being quantified and certified by the WHO. So the areas in which there is a high transmission rate, the government generally focuses upon the distribution of these bed nets. So they have played a very important role in decreasing the number of cases of malaria in those particular area. The next one is called as the indoor residual spraying with the product pre-qualified by the WHO. So there are various indoor residual sprayers which are available, which are called as the IRS. So these are sprayed across the areas where there is a probability that the mosquito might be breeding or settling down. Now, the malaria preventive chemotherapies have been broadly classified into two major types. One is called as the chemo prevention, the other one is called as the chemo prophylaxis. Now, we have to understand what exactly both of them, then only we can understand the antimalarial drug resistance. Now, the chemo prevention, as the name is suggesting, it means that it is a full therapeutic course of antimalarial medicines at a pre-scheduled times. So it is irrespective of the infection status, and it helps to prevent and neutralize all the parasites from that particular patient. So the chemo prevention is given to the patients actually, while the chemo prophylaxis mainly involves the administration of a subtherapeutic doses of antimalarial, which are majorly taken by the people who are traveling to the malaria endemic areas, so that they can prevent themselves from any new infection. So chemo prophylaxis is a part of the stopping the malarial infection in the new persons who are traveling to the malaria endemic areas. Now this is a diagram which is showing the various parts of the diagnosis as well as the treatment regions. Now if we see over here, the diagnosis is majorly by the microscopy, then The next level, advanced level comes are the ELISA kits which are available, the PCR techniques are available which have now gone into the qPCR or the quantitative PCR. Real-time PCRs have also come into the picture. While if we go to the antimalarial drugs, there are various antimalarial drugs which are available in the market, which has been classified into various classes. And finally, there are novel antimalarial drugs which are under the process of clinical trials, and over the years they may come into the market once they are approved by the WHO. Now, the malarial parasites are identified by a very basic but a gold standard protocol which is called as the microscopic diagnosis. The malarial parasites are identified under the microscope, in which the patient's blood is taken, then a smear is being made which is called as the blood smear, and finally it is being stained with a staining technique. Majorly the Giemsa stains are being taken. So these Giemsa stain gives a coloration to the parasite DNA which distinctly appear under the microscope. Now, the microscopic diagnosis plays a very important role in the identification, especially during the field studies. Now, if you see the picture over here, this picture shows the microscopic images of the malarial parasites taken from a patient. So we can see on the left hand side picture, these are the RBCs which have been stained, and inside the RBC, if we can see, the parasite infected RBCs are also shown over there. So the right hand side image shows one of the erythrocytic stages which is called as the ring stage, while in this particular image, if we see in the center, there is a trophozoite stage, and on the top of the trophozoite stage there is a scission stage which is also available. So these are the various stages which are present in the red blood cells, and they infect the red blood cell, and finally when the scission stage is released, they rupture out the RBCs. Now, the scissions which are being released can go and freshly attack the new red blood cells in the blood circulation. So that's called as the erythrocytic cycle of the malarial parasites within the host system. The next important diagnosis measure is the antigen detection. So in the antigen detection, one of the major detecting kits is called as the rapid diagnostic kits. Now, these rapid diagnostic kits are basically based on the technique of ELISA, which is the enzyme linked immunosorbent assay. Now, these rapid diagnostic kits are often useful as an alternative to the microscopy. So they help in checking out whether the microscopic results are positive or they are false positive. So they help to check the microscopic results actually. Now, how it is being done? Now inside the cassettes of these rapid diagnostic kits, there is a strip which is made up of the filter paper and a nitrocellulose is present. Now, a blood drop is put over there in the RDT kits. So in one of the hole, and from the other hole a buffer is being added. Now, these buffer are basically the lysating agents which are there. So the buffer carries the blood along the length of the RDT, and what happens is, over the spread there are presence of the lysing agents which is present in the test strip. So what this lysing agent does is, they ruptures the red blood cells, releasing the parasite proteins over there. Now, once the parasite proteins are released, there are presence of dye-labeled antibodies which are present. Now, what these dye-labeled antibody does is, they go and bind to the parasite antigen over there. Now, once this dye-labeled antibody binds to these antigens, these targeted antigens then further moved at the end of the nitrocellular strip. Now, as they start moving into the nitrocellular strip, at the test region there are presence of antibody which are specific to these antigens. So they are bound at that particular region. As soon as these dye-labeled antibody along with the parasite antigen reaches those regions, what happens is, in the test well, as there is a presence of already bound antibody, so there is a complex which is formed. So we can see in the image over here, we can see there is a bound antibody to the nitrocellulose membrane, to which the antigen is bound, and to this antigen there is a addition of a dye labeled antibody which is there. Now this forms a sandwich ELISA kind of reaction, and the dye which is present, what happens is it gives a coloration in those particular test wells. And if the patient is having a infection to that particular species of malaria, for example Plasmodium falciparum, in the test well there will be a chances that the test well will be shining over there with that particular coloration. So the test well will give a coloration, and that shows that the person is positive for that particular infection condition. Now this is the test which is being done. So these RDTs are the one which is being used. So if the antigen is present over there, some labeled antibody antigen complex reaction occurs, which I just showed you in the previous slide, and what happens is if it is a positive result, they will accumulate on the test line, and once the accumulation occurs, what happens is they will start coming up and giving up in those particular test line. Today I have brought a RDT kit for my viewers to see how exactly these RDT kits work. So if you see over here, there are two wells over there. So one is for the blood, the other one is for the buffer. As it moves down, if you see over there, there are different test and the control wells which is there. So this is a RDT kit which can test simultaneously two species, Plasmodium falciparum and Plasmodium vivax over there. So as the test well indicates over there, so if you see over here, the Plasmodium vivax test well is being shown over here. So it shows that the patient was positive for the Plasmodium vivax while he or she was negative for Plasmodium falciparum. So this is a very important antigen detection kit which has been developed and which can functions in a very short span of time and can help you detect what exactly the infection is and the person is suffering from which particular kind of species of infection. Now apart from these antigen detection, there are various molecular diagnostic techniques which are available, which includes the basics of the polymerase chain reaction. We know that the polymerase chain reactions helps to amplify a specific sequence of the DNA. So if we know which sequence of the DNA is present in the malarial parasite, we can easily quantify and amplify and check it in these gel runs over there. So the PCR technique is one of the most useful technique which is helping the scientists across the globe to confirm the species of the malarial parasite, because they are very specific and very targeted in their approach and help to establish the results which are being given by the smear microscopy or the RDTs. Now this is a timeline which is showing how exactly the work process of a molecular diagnostics happens. So we know that there are various qPCR, which is a quantitative PCR platforms which are available, which are portable, cost effective and can be easily deployed in the field so that a real-time images and real-time results can be detected in those particular fields. So first the samples are being isolated, the blood samples either from the patients or the mosquitoes are being taken, then the DNA is being isolated from them. There are various kits which are available, and finally the PCR is being run, which can run in a short span and amplify those particular specific region, and finally the results outputs are given which are easily quantified with the help of the maps over there. So this is one of the molecular diagnostics techniques which is being available, which helps the scientists to quantify and check what are the various species infections which are present in those particular patients and how exactly the treatment regions can go into those particular regions. Now in this part of the lecture, what we discussed is the basics of the malaria, and in this malaria we discussed about the vectors which are present in the malarial cases, the Anopheles vectors which are available, the various species, the life cycle of the Anopheles mosquitoes, and then we discussed about the diagnosis and the treatment regimes which are available for these antimalarials. With this note, I come to the end of the first part of the lecture. Thank you very much.